What is a haplogroup in DNA, and why are haplogroups in DNA important? Remember, part of this channel's goal is to figure out who are the ancient Hebrews of Israel, what is their genome, and who are their modern descendants today. First of all, we need to know and keep in mind that the Israelites are a Semitic people, and according to their genealogy, they descend from Shem. This is according to the male lineage, the male line. Basically, Shem is their paternal ancestor. But why is this important? With that being said, we must look at the origins of Afroasiatic and Semitic peoples, specifically Proto-Afroasiatic and Proto-Semitic. Where is their geographical homeland, and what time period did they come into existence? These are the methods we can use to determine the genome of ancient and modern Israel. One of the best tools we can use to figure this out is Y-DNA haplogroups. Now, disclaimer. For the videos and information that I'm about to use and share, I am protected under the Fair Use Act. This is for teaching purposes, research, and commentary. And we're going to look at a quick video on what is a haplogroup.
As many Americans know, the third President of the United States and author of the Declaration of Independence, Thomas Jefferson, was an important figure in the emergence of early American identity and is considered one of the major founding fathers of the country. And although many today argue over his own personal moral character and ethics, it was a major point of contention even hundreds of years ago, and many spread rumors of Jefferson's ongoing affair with his slave Sally Hemings and whether he was the father of her 6 children, an act which would have been highly frowned upon even in the 18th century. Centuries later, in 1998, it has seemingly been proven through DNA tests that Jefferson was indeed the biological father of Henning's children, despite the fact that there were no viable biological samples of the deceased president in existence. So how did they do it? When it comes to human genetics, perhaps the best way to determine hypodescent or common ancestry in individuals and population clusters is through a specific genetic marker known as a haplogroup. Haplogroups are a common way for geneticists, archaeologists, and historians to make conclusions over the origins, exchange, and evolution of people groups all over the world, and you may have heard me mention them in videos from time to time for the past year, but unless you knew beforehand, information on haplogroups may just cause confusion and misinformation for many viewers. Therefore, I've created this video to better give an understanding of how haplogroups relate to genetics, race, ethnicity, community, and other aspects of humanity that paint such a beautiful picture of the history and diversity of our planet. For as long as can be traced back, all females carry a maternal haplogroup inherited from their mother, while males carry both a maternal and paternal haplogroup, seeing how men have an X and Y chromosome. The paternal haplogroup inherited by the father is known as the Y haplogroup from the Y chromosome, while the maternal haplogroup is inherited from mitochondrial DNA and is thus referred to as the mtDNA. Because of historical patterns of migration and the nature of male-female relationships for most of human history, generally speaking, paternal haplogroups are better studied, understood, and mapped out, although this doesn't make maternal lineage irrelevant or any less interesting. For this reason, haplogroups will only survive to the next generation if they're passed on to their offspring of the same sex through the X or Y chromosomes. Now, you may not necessarily ever have an offspring that is the same gender as yourself, in which case your specific haplogroup would die with either you or your child, but at least one of your parents sure did, as with one of their parents and grandparents and great-grandparents and so on, back to the genesis of the human species. Barring genetic mutations, if you are male, it's an absolute certainty that you will share the same paternal haplogroup with your father, all of your biological brothers born of the same father, all of your paternal uncles, any paternal male cousins born to your uncle, and of course your paternal grandfather, his father, and so on, and all biological male children of yours will share your paternal haplogroup. Similarly, barring genetic mutation, regardless of gender, you will share the same maternal haplogroup with your mother, all of your biological siblings born to the same mother, all of your maternal aunts and uncles, any maternal cousins born to your aunt, as well as your maternal grandmother, her mother, and so on. And if you are female, then all biological children of yours will share your maternal haplogroup, although only your female children will pass it on to their offspring.
So as we can see, why DNA haplogroups are a perfect tool in discovering the paternal ancestry of Israel's ancient ancestors, and of course, this can help us find the descendants of Israel today. With that being said, we will use more DNA methods to determine who are the ancient Israelites. Therefore, let's get a basic understanding of DNA. Again, disclaimer, I'm going to play a video clip that will be breaking down DNA and what it is. So this is for teaching purposes and educational purposes and research. I am protected under the copyright law and the fair use law. So, what is DNA?
What is DNA? Every living thing has DNA, from plants to animals to bacteria, even to viruses which were once considered non-living. And most importantly, we humans have it. DNA stands for deoxyribonucleic acid, and although every living thing has DNA, its varied compositions differentiate you from others. The interesting fact is that humans' DNA is 99% the same. Therefore, it is only 1% of DNA that makes you unique. Does this mean twins don't have exactly the same DNA? No, they don't, unless they're monozygotic or identical twins. So now the questions arise. Why every living thing has DNA? What is so important in it? And what does it do? DNA has a recipe for synthesizing proteins. DNA synthesizes RNA, or ribonucleic acid, by a process called transcription. It is further translated into proteins by a process termed as translation. The explanation of DNA conversion into RNA, and finally RNA into proteins, is called central dogma. DNA contains hereditary information that is passed on to you from your parents. If you and your father have the same colour of eyes, or your hair are as silky as your mother, it is because of DNA that you inherited from your parents. Even many diseases are passed on from generation to generation because of DNA. Where does DNA reside in your body? Your body is made up of billions and trillions of cells, and almost all cells have DNA in their nucleus. Can you tell us which cell may not contain DNA? Now let's talk about the structure of DNA. Think of DNA structure as a ladder whose rungs are made up of different bases and sides as sugar phosphate. Each composition of this nitrogenous base, pentose sugar, and phosphate group is called nucleotide. The nitrogenous bases are of 4 different types: adenine, guanine, cytosine, and thymine. Adenine will always bind with thymine, and cytosine with guanine. This type of bonding is hydrogen bonding. Phosphate and sugar are linked together to make backbone, and complementary bases are attached on it. Due to the bond angle of the sugar-phosphate molecules, the linkages will eventually form a double helix structure. There's a lot more to know about DNA, but that's all for now.
So now that we have an understanding of what is DNA, Now let's look into genes. What are genes?
Every person in the world is different. We all have physical characteristics and quirks that make us unique. But what biologically makes us who we are? Our genes. Genes are found in every one of the 37 trillion cells that make up our body. To define what a gene is, let's look closer at one of those cells. Every cell has a nucleus, and inside every nucleus are chromosomes. And all chromosomes are made of DNA, molecules that contain genetic information called genes. Genes in general direct specific processes in the body by encoding for proteins. A gene's primary function can be broken down into 4 general categories. First, genes can encode proteins to build something in the body. This can be anything from muscle to bones and cartilage, and any other of our internal structures. Secondly, genes can encode proteins that help direct the body to do something, such as muscles flexing or hair growing. Thirdly, genes encode proteins that indirectly support bodily functions. This could be by helping the immune system to respond to injury or to assist blood flow through our circulatory system. And lastly, genes directly influence our physical characteristics or traits like physical quirks, hair color, eye color, skin tone, and more. As you can see, each unique combination of genetic code is responsible for everything in our bodies, from internal operations to the way we look. With 20,000 to 30,000 genes in the human body, understanding the function of each gene and even understanding how different genes work together continues to be a major topic of research. So to put it basically, one last time, genes encode proteins to direct specific purposes in the body.
Alright, so we have a basic understanding of what genes are. Now let's dig into chromosomes. What are chromosomes?
Chromosomes. In the nucleus of each eukaryotic cell, the DNA is packed together into chromosomes. Each chromosome is made up of DNA that is tightly coiled around proteins that give it the structure. This tight packaging system means that 2 meters worth of human DNA can fit into a tiny cell that is only 2 micrometers wide. In this video, we are going to look at what chromosomes are and what their role is. Chromosomes usually occur in pairs, except for gametes, which are eggs and sperm cells. But the number of chromosomes changes depending upon species. Humans have 23 pairs, elephants have 28 pairs, and carrot plants have 9 pairs. If you want to learn more about DNA, watch this video. So chromosomes are made from long, coiled molecules of DNA known as double helix. Within this long chain, there are shorter regions that carry the genetic code for particular proteins that are known as genes. Chromosomes carry all of the information that help a cell grow, survive, and reproduce. Each chromosome has a centromere, which divides the chromosome into 2 sections, or arms. The location of the centromere on each chromosome is what gives it its characteristic shape. The P arm are the short arms, and the Q arms are the long arm structure. Chromosomes are passed from parents to offspring and play an important role that ensures DNA is copied and distributed accurately in the process of cell division. Chromosomes are not usually visible in the cell's nucleus and can only be seen when the cell is dividing. During cell division, the chromosomes are duplicated. We will look at this in more detail in future videos, such as what is mitosis. When the chromosomes are duplicated, they condense into short structures which can then be strained and observed under a microscope. The centromere holds the duplicated chromosomes together. Duplicated chromosomes are commonly called sister chromatids. So, from this video, you should know that chromosomes hold the DNA inside the nucleus. They have a centromere which holds the duplicate chromosomes together, and these duplicates are known as chromatids. Humans have 23 pairs of chromosomes, but other species have different amounts.
What is a chromosome? To solve problems that explain and predict traits and variations, we first have to understand cells. All living organisms are composed of cells. Cells work like little factories doing all the jobs inside your body that are needed to keep your body functioning. Your body is made up of many different kinds of cells, such as skin cells, muscle cells, and nerve cells. Some cells look like squashed bricks, some look like donuts, and many have irregular shapes. However, every cell, no matter what its job, has the same basic parts. All human cells have an outer border that's the boundary of the cell, called the cell membrane. A liquid material called cytoplasm is inside the cell membrane, and there's a large structure suspended in the cytoplasm called the nucleus. The nucleus is the part of a cell that contains the genetic information. The nucleus is surrounded by a nuclear membrane that, like the cell membrane, makes a boundary around the nucleus. Now let's explore chromosomes. Chromosomes are thread-like structures located inside the nucleus of animal and plant cells. Each chromosome is made of protein and a single molecule of deoxyribonucleic acid, or DNA. Passed from parents to offspring, DNA contains the specific instructions that make each type of living creature unique. The term chromosome comes from the Greek words for color, chroma, and body, soma. Scientists gave this name to chromosomes because they are cell structures, or bodies, that are strongly stained by some colorful dyes used in research. So what do chromosomes actually do? The unique structure of chromosomes keeps DNA tightly wrapped around spool-like proteins called histones. Without such packaging, DNA molecules would be too long to fit inside cells. For an organism to grow and function properly, cells must constantly divide to produce new cells to replace old, worn-out cells. During cell division, it's essential that DNA remains intact and evenly distributed among cells. Chromosomes are a key part of the process that ensures DNA is accurately copied and distributed in the vast majority of cell divisions.
So we have a pretty good understanding of what chromosomes are. Now let's look into STR analysis. What are STR analysis? What is that?
A short tandem repeat, or STR, is a region of DNA composed of a short sequence of nucleotides repeated many times. Since the number of repeated sequences in a given STR varies from person to person, pinpointing these variants can be useful in DNA fingerprinting. As the name implies, the repeated stretches in STRs are short, only 2 to 10 base pairs long. For this reason, and because they are dispersed more evenly throughout the genome than the longer variable number tandem repeats, or VNTRs, STRs are favored by forensic labs. Large databases of information on STRs in the general population tell analysts how much variation exists at any given STR location. That information can help forensic analysts determine the conclusiveness of a match between two samples. If 10% of people have the same number of repeats at a given STR, for example, finding a match at that site is merely suggestive that two samples are a match. If two sites with the same prevalence match, the odds drop to 1 in 100 that the similarity is coincidental. If 13 sites match, the odds that any two people would possess such a fingerprint are so small, about 1 in 10 trillion, that the result can be considered a definitive match.
So now we know what an STR analysis is. Now let's look into autosomal DNA. What is autosomal DNA?
Welcome to the Ancestral Findings Podcast. Have you considered doing an autosomal DNA test to add to and improve the accuracy of your family tree? Have you even heard of an autosomal DNA? It is the most common type of publicly available DNA test nowadays and is quite affordable. Here is what you need to know about this valuable genealogical DNA test. Are you familiar with autosomal DNA? If you're not, you should be. It is one of the most important and common ways of using DNA to add to your family tree and to improve its accuracy. Now is the time to learn about autosomal DNA and what it can do for you in your genealogical research. While Y-DNA traces only ancestors on the male line of your family, and mtDNA traces only ancestors on the female line, autosomal DNA traces both sides equally. There are parts of our DNA that researchers have not yet discovered a purpose for, and this DNA was commonly referred to as junk DNA in the past. However, genealogy experts soon discovered that this so-called junk DNA could be used to reveal family relationships for both men and women. This was an improvement on the Y-DNA test that only men could use, and the mtDNA test that traced females who usually change their last names upon marriage. Anyone in your family can take an autosomal DNA test. It will also show you people who are related to you on both sides of your family. Most DNA testing companies today do only autosomal DNA testing, as it is the most useful type of testing for the most number of potential customers. A few companies still do Y-DNA and mDNA tests for those who need those tests particularly, but autosomal DNA testing is still the most commonly used test. It is also highly affordable because it is used so much by so many customers. When you take an autosomal DNA test, the testing company will show you matches to other people in their database who have also taken the test. These matches may be on your father or mother's side of your family. In most cases, you will have to contact the ones who have agreed to allow contact from their matches and discuss your family trees to determine which side they fall on. Sometimes it will be obvious because of a particular surname, or because you see they are also matched with someone you already know. If you can get one or both of your parents, grandparents, or anyone who is a sample from both sides of your family, you will get much more accurate results, and some sites, like 23andMe, will actually be able to tell you which side of the family that person matches on. The more of your relatives who test, the more accurate your results will be for your matches. When you meet your matches online, you have a very good chance of learning new family tree information you previously did not know. You might even find someone who has a family Bible or old family photographs and who is willing to share them with you. Basically, when you have your autosomal DNA tested, you are able to find out much more about your entire family than with any other type of DNA test. It is worth it for any genealogist to have this test done.
So now we have a proper understanding of what autosomal DNA is. Now the last DNA method that we should know about is LL-DNA. So what is an LL-DNA?
Hello, you are watching Quick Definitions, stated clearly. What is an allele in genetics? As we've learned in earlier videos, a gene is a stretch of DNA, usually a long stretch of DNA, that codes for something. What exactly do genes code for? Technically, they either code for specific proteins or they act as templates for functional chains of RNA, but because proteins and chains of RNA go on to produce traits, things like eye color and bone length, we can say that an organism's genes, in a roundabout way, code for an organism's traits. An allele is an alternative version of a specific gene. If that sounds confusing, that's okay. It should become clear once we see a simplified example. Let's say there is a gene in a species of frog that codes for the frog's eye color. We will call it the eye color gene. No need to get too creative here. This frog has a version of the eye color gene, an allele of the eye color gene, that codes for yellow eyes. He has the yellow allele of the eye color gene. His buddy here has a different allele, a different version of the eye color gene. He has the red allele of the eye color gene. Now, this hypothetical example is overly simplistic. In real life, there are multiple different genes and sometimes environmental factors that all play together to determine an animal's eye color. Also, most animals have 2 copies of their own DNA, meaning that a single animal can actually have 2 different alleles or 2 different versions of any given gene in its own genome. Furthermore, most alleles don't have any actual effect at all on a creature's observable traits, but don't worry about any of these technicalities for now. The important thing to understand is this: an allele is an alternative version of a specific gene. That's it. New alleles, new versions of old genes, come into existence via mutations. Once a new allele exists, it is forced to compete with other alleles in the population. For example, when the yellow eye allele happened to emerge within our hypothetical frog species, it might have given its host a survival advantage because its host could better blend into its environment and hide from predators. Because of this, over many, many generations, the yellow allele could dominate the population, completely outcompeting the red allele, causing the red allele's extinction. When an allele dominates an entire population, we call this fixation. So there you have it. An allele is an alternative version of a specific gene. I'm John Perry, and that was a quick definition of an allele stated clearly. Well, what do you know? There's brand new Stated Clearly t-shirts for sale. I think I'll buy myself 2 of them.
Within the typical body cell of an organism is a set of chromosomes, and in humans there are 46 chromosomes. We get 23 from the mother and 23 from the father. Now what this means is that for every single 1 chromosome from the mother's side, it matches with 1 chromosome from the father's side. This gives you 23 homologous pairs of chromosomes. As for the chromosomes themselves, the chromosomes are just highly organized structures. And in these organized structures are tight coils of DNA. And that's because DNA is a really large molecule. As for the DNA itself, you can see that it has the shape of a double helix. A double helix is like a ladder that's been twisted, and the rungs of those ladders are made of something called base pairs. As for the bases themselves, we get 4 types of bases. You've got A for adenine, T for thymine, G for guanine, and C for cytosine. Notice that in the structure of DNA, A only ever goes with T, and G only goes with C. Now, as for how genes fit into all this, genes are actually just specific lengths of DNA that code for characteristics such as eye color. So this length of DNA here could represent the eye color for something, for say, blue eyes. And over on the other homologous chromosome, on the other matching chromosome, that same section, that same gene could be something for, say, brown eyes. Now here's the catch. For this to represent blue eyes and the other one to represent, say, brown eyes, this means you have different forms of a gene, and different forms of that same gene are called alleles. So this could be the allele for blue eyes, and this would be the allele for brown eyes. Now, the only differences between the two are that, for the most part, the order of the bases are actually the same, but there's only a slight difference in the order of those bases.
All right, and I just want to make it clear once again that I am protected under the Fair Use Act, and I use these videos for educational purposes, for teaching, and again, I am protected. Okay, so now that we have a proper understanding of Y-DNA haplogroups, what is Y-DNA, what are genes, chromosomes, STR, autosomal DNA, and Allele DNA, we're going to use all these methods to figure out who are the ancestors of Israel before Abraham, Isaac, and Jacob, which are Proto-Semites. We will use all these methods to discover the genome of the ancient Hebrews of Israel themselves, which in all should give us a proper understanding as to who their descendants are today. So we're going to use all these tools to figure this mystery out. And with that being said, we will also use these methods not only for the research of Israel, but also to understand who were the ancient neighbors of Israel, the other ancient civilizations and people groups that lived in the ancient Near East. Because understanding Israel is cool and interesting and good for our research, but it is also very important to know who are the surrounding nations of Israel, who are these other ethnic groups that lived in the ancient Near East, because this can give us a proper understanding of who Israel was dealing with, who Israel could have mixed with, who could have invaded Israel, and things of that nature. So we'll use DNA both for Israel and their neighbors. But with that being said, I believe that DNA is a good tool that we can use. And DNA is something created by the Most High, or by God, by the Creator, because it is within us. It is a perfect map. It is a perfect, magnificent thing given to us that can show us who we are going back thousands of years. And I don't think this was placed in us on accident. It's a code. DNA is a code that can lead us backwards in time. And it's actually amazing how how magnificent and how complex it is, because DNA, it's something out of this world that the Most High gave us. And not just us, but also animals and plants, because everything has DNA. I don't believe DNA is junk science. I believe this is something that we should use. And this is where we end the video on DNA and why I'll be using it and what are the methods we will be using. And I hope you enjoyed it, and I hope you join me on our journey on the search of Israel as well as their neighbors, and when it comes to the scriptures, history, and archaeology in the four fields of anthropology. Thank you, and have a great day.